Thank you all for being here so late in the afternoon today. I would like to first thank the people that nominated me for this award, and I'm very honored to stand here. So the Peter Kilham Memorial Award was awarded to me, and of course I read his papers some years ago, but now that I have received this award, I read his papers again. I never had the pleasure to actually meet him, but I'm sure that if we would have met, we had much to talk about. He also looked a lot in comparison between temperate and tropical systems, which I personally also really like, and he had a lot of biogeochemistry work that is also very interesting to me. So I'm very honored to stand here in front of you talking about the work that we have been doing. So I am standing here, but actually the work that I'm going to show you is based on group work together with a lot of different people from a lot of different institutes around the world, particularly though in the Netherlands and in Brazil. So in my talk I'm going to shift a little bit back and forward between the Netherlands, where I'm from, and Brazil, where a lot of our work is being conducted. So we work together with universities, but also with consultancy companies and with different water authorities. And of course, I would also like to thank all the sponsors for the work that we were able to do. So in my talk, I want to go through different topics. I want to very briefly introduce the global importance of greenhouse gas emissions, aquatic greenhouse gas emissions. Talk a little bit about different emission pathways and their quantification. And then I'll go into the biotic and abiotic drivers of these greenhouse gas emissions and their importance on a landscape scale. Then the final part of my talk is about mitigation measures of these aquatic emissions. So first, the global importance. I'm going to talk mostly about methane in this talk, but when we think about aquatic greenhouse gas emissions, of course, there are also other very important gases, CO2 and N2O. I will explain you in a little bit why the focus on methane. Well, methane is important, it's a very important greenhouse gas. It's responsible for roughly 30% of our global warming currently, and wet areas are very important sources. And I think most of you have seen this figure before that shows very clearly that there are a lot of anthropogenic methane sources, but also that aquatic systems are very important. And very often we then focus on where I put this red square, and we kind of forget that there is also a very important aquatic component in this part. So I think many people realize that there's a lot of emissions from rice fields, but there's also a lot of methane emissions from aquaculture. And that's one of the things I will touch upon in my talk as well. So I will focus also on how we can mitigate these emissions. And when I talk about mitigation of aquatic greenhouse gases, of course, I'm not talking about the natural emissions that come from our natural systems, but I really focus on the anthropogenic methane emissions from aquatic systems. And these, according to the IPCC, are emissions from human-made water bodies, such as aquacultural ponds, but also enhanced emissions due to alterations of natural systems by human activities. Think about eutrophication, for instance. So when we talk about emission pathways, it's very well known that if the concentration in the water is higher than in the atmosphere, there is diffusion out of the water into the atmosphere. When there's a lot of primary production, the CO2 flux can also go inward, and we get an uptake of CO2. For methane, however, methane dissolves very badly in water and is heavily produced in anaerobic conditions in the sediment. It dissolves very badly, and there bubbles can also be formed. I also had an N2O flux there. And these bubbles very episodically rise to the surface, for instance, when there's The buoyancy there's very high buoyancy, so the bubble comes up, but also when a large low pressure front passes, the bubble expands, it liberates from the sediment and rises to the surface. So this does not happen continuously, but is a very episodic event, which also makes it more tricky to accurately measure it. And abolition from shallow systems, and most of the systems that I work in are shallow, is a very important emission pathway. Most of the time it takes up more than 60% of the total methane emissions that we measure. So it's very important to quantify it in a good way. So here I have a small video showing how these bubbles look like. We are all aquatic ecologists, so we have all seen this. But this is in a Dutch pond where we arrived, and it looks like a bubble bath, you know. And you think, oh, this is a hot spot, and by the time that you take the measurements equipment out, the bubbles are gone. Here I disturb one of our ditches. The previous picture was in a natural area. As far as in the Netherlands, we have very pristine areas, but that, on a side note, this is a very eutrophic ditch. And when you see that there are really a lot of bubbles coming out of this. And actually there was one artist inspired near our university that is catching these bubbles, and he drives his motorcycle now on bubbles that he's harvesting from these ditches, because it's also a fuel. So we have a lot of bubbles. So I've been discussing that we have the diffusive flux, we have this bubble flux, and there's also a third flux, and this is the emission pathway through plants. And I will also be talking about that later. So how do we quantify these emissions? We work a lot with floating chambers that we put on top of a water surface, and then we connect it to a greenhouse gas analyzer, and then depending on the analyzer that we connect, we can measure CO2, methane, and sometimes also N2O. We have bubble traps, which are kind of funnels that we hang in the water of various sizes, very large ones for deeper systems, but our challenge is also to have very shallow funnels, because we work also in these ditch systems that are sometimes only 10 centimeter deep, or sometimes even less, and they bubble a lot as well. We have a lot of mesocosms, so we also do a lot of experimental work, but we also do a lot of work in outdoor real systems. So our group has quite a lot of these terrestrial automatic chambers that open and close, and you can buy them very easily in different manufacturers. That doesn't mean it's not a challenge to keep them running, because it really is. And now we also have a floating chamber that can automatically open and close. So we are very proud of this award-winning device that one of our colleagues developed together with our Techno Center. And the idea is that we can leave these standalone in different systems, and they close and they open, and we can have very high frequent diffusion and ablative fluxes in different sites. And that is really, I think, a very important advancement, because nowadays, the data that we have is mostly daytime measurements. We know very little about dial variation, and also temporal variation. You know? When it's very bad weather, we are most of the time not there, and then the highest fluxes may occur. So this is something we're going to employ as soon as we are back in the Netherlands. So it's a challenge to actually really quantify fluxes well, and I think with the common effort of all our colleagues around the world developing new devices, we are getting better and better at it. But of course, what also really interested us is what is driving these emissions. And here we look at different biotic and abiotic drivers. And we know that temperature is a very important driver of methane emissions. Of course, also substrate availability. When there is no substrate for the methanogens, there will be no methane produced. The availability of terminal electron acceptors is very important, and of course also the microbial community. So these are all things that we look at. So I'll start with the temperature. I think Some years ago, we did like a meta-analysis on all the data that was available on methane emissions and temperature that was out there. And we found a very strong exponential increase in methane abolition with temperature. This led to the insight that with each degree of warming, we have 6 to 20% more methane emissions from our systems. 6 to 20%, that is really a lot. In the waters that we work in the Netherlands, the temperature has already risen 2 degrees over the last 3 decades. So we are already in a much higher methane emissions than in the previous periods. Also in this experiment that we conducted in these highly controlled mesocosms that are situated at the NEO Institute in Wageningen, we found the same effect. When it's warmer, it increases. And of course here we also had like a seasonal effect, but also when we have a control and a warm condition, we both had a seasonal effect, but one 4 degrees warmer than the other, we found a very similar effect of warming. So the warmer, the more methane. The warmer, the more methane, but the more nutrients, so the more eutrophication advances in the system, also the more greenhouse gases are being emitted. So here I put the ablative flux of an experiment that we did some time ago, where we had different nutrient loadings to mesocosm systems. And in these systems, we also had different plant species. So I listed the species here, and if you don't know them, the blue ones, they are not rooted in the sediment, and the ones that are depicted in red, they are typically rooted in the sediment. And although we see a lot of scattering, because we had some herbivory occurring in the systems as well, you see really that the red dots seem to be on the lower end. Of course, the overall picture is that the more eutrophic, the more greenhouse gases we found. And this is a really strong effect because it's, as you can see, it's a log scale on the y-axis. It also comes with kind of a good message, because if we would manage to reduce this nutrient loading, of course this is already a mitigation strategy, we could really reduce the greenhouse gas emissions. And this is an example of a win-win situation where we would get better water quality and also less greenhouse gas emissions. And this goes for methane, but the same, I did not show it here, goes for N2O emissions, which also skyrocketed in this high nutrient loading. So particularly when we have rooted macrophytes, so macrophytes, plants that root in the sediment, we found lowest emissions. So this made us dive a little bit more in the role that different types of water plants play in methane dynamics in systems. So together with Pascal Bootmer, you see in the picture here, and a group of other people, we wrote a review on the different roles that plants play when it comes to methane emissions. So of course they're important producers of organic matter, and in that sense they could also stimulate methane production. But they are also very rich in microbial community on their leaves, their stems and the roots, where they provide a niche for methanotrophs. So they also provide niches for methane consumption. As I already told you, they can also transport methane through the roots and the stems to the atmosphere. So emergent plants can also play a role in emitting greenhouse gases. So what we believe that we saw in this experiment that I showed you before, where the rooted plants had lowered the methane emissions, is that these were plants that typically leak a lot of oxygen through their roots. So the rhizosphere is kind of aerated, which then blocks methane production potentially, and could also enhance methane consumption. So in these both ways, it reduces the methane emission to the water column, and hence also to the atmosphere. We saw this also in this indoor experiment that I talked to you about later. So this the orange curve we already saw before. This was an algae-dominated system where we've seen a very clear increase in methane abolition with warming. If we would have the same systems with duckweed, so a small floating plant, we would find much higher emissions than with the algae. And this was related to the very low oxygen concentrations below this floating mat. There's no light penetrating this mat, so there's no primary production. And it's also very hard for oxygen to diffuse into the water through this dense mat of macrophytes. So you get an anaerobic situation with a lot of organic matter being produced by these plants that sinks down, which really fuels the methane production in these systems. The very bottom line is our treatment with submerged plants. So again, this is a plant, Myriophyllum spicatum, that leaks a lot of oxygen to the sediment, and this really reduces the methane emission from the systems that we were working with. So we have a lower emission and also a lower temperature response. Unfortunately, and this is an update from a paper that was led by Brian Moss over a decade ago. We know that climate change favors floating plants at the expense of submerged plants through a variety of measures. So Mariana Meerhoff, also present here, she led this work on this review together with scientists from all over the globe, I would say, where we combined the evidence that we see that with warming, but also changes in precipitation and coinciding in enhanced incidences of fires, really increases the nutrient loading to our systems, which then favors plants that do not have access to the sediment, but profit from the high nutrient concentrations in the water. So this then also leads to higher methane emissions, as we have just seen. So it's a very strong feedback between climate change. It gets warmer, we get more floating plants, we get more emissions, hence more floating plants. So that's a very strong positive feedback, although it works out negatively for us. But that kind of brings me back to where I started when I was talking about Kilham, comparing the tropical and the temperate systems. So what we found in our temperate systems is that this duckweed invasion would really trigger the methane emissions from the systems that we were working at. But we were really wondering if the same principles would work also for tropical systems, and then not these little tiny plants that we have, but working with this large water hyacinth plant that you probably all know with this nice purple flower. It's very invasive in many species of the world, but here in Brazil, it's a natural occurrence, at least in natural systems. Of course, it's also a problem in very eutrophic systems where the flow is blocked, like in some reservoirs, where it also has to be removed regularly. So we went out, and I say we, but this was mostly the work done by Hernandez, who is also here. We went to 22 places that we sampled twice, in the Pantanal and in the Amazon, and we looked at the greenhouse gas emissions from these floating beds and also outside these floating beds. We also had our bubble traps, and what is very special here is that we put these floating beds in between the macrophytes, but we also made sure that the roots that we detached from the rest of the plant were still below these funnels, because we were interested in knowing if these roots would trap the bubbles and avoid them from coming up. So indeed, this was kind of what we were looking for. Do these plants block the bubbles? For these large plants, it's difficult to see, but we really saw that for our duckweed systems to really happen. And when they are blocked, so then when the bubbles stay there, they can dissolve again and potentially be oxidized in the water column. So we measured this inside the beds and outside the beds. And what we found, of course, is very scattery. But what we found in general is that when you have a higher biomass of water hyacinths, so when you go to the right on the x-axis, you find a relatively low abolition compared with the open water. So this kind of indicates indeed that some of these bubbles are trapped in the rhizosphere of these large bubbles. What was very interesting too is that the diffusive flux, which in this case also includes the emission through the plants, because we put our chambers on top of the plants, did not vary much between the open water and the plant bed. And that is very much contrasting what we found in this temperate system. What was really eye-opening for me was the incredibly low oxygen concentrations in these open waters, because most of them were below 3 milligrams per liter. So these are very organic, rich rivers that are also extremely hot. Very nice to swim in, by the way, if you avoid the piranhas. And they had already high methane diffusion from the open water. So there was not a lower oxygen concentration below the plant bed. And the methane diffusion was therefore very comparable between these two sites. Yeah. What was also clear is that the diffusive flux, again, this also includes the plant-mediated emission, was much higher when these huge water hyacinths, which can have roots more than 1 meter long, reach the sediment. So some of these floating plants are actually also rooting in the sediment when the water level is sufficiently low. So here we just clustered the emissions from the plants that were actually having access to the sediment and those that were free-floating, and you can really see the difference. The unexplained variance is still very large, of course, but we attribute this enhanced flux really to this plant-mediated emission, when the roots could tap into this very oxygen-rich pore water in the sediment. So that is what we see here. When we have these rooted plants, we found much higher emissions, but when they were floating, we did not find a very clear difference in the methane emission between the open water and the plant bed. We were not so satisfied with this huge variability that we could not explain, and that's why we decided to do also a laboratory experiment. I think we are probably the last people in the Netherlands that have a permit to have these plants, because they're currently on the forbidden list to be sold or to be kept, because they're so invasive. We cannot have them anymore in Europe, but we still had some legally. And we could do some experiments with them, and these were conducted by Quinten Struik, who's also present here. So if you want to hear more details, please talk to him later. So we put the plant on an aquarium, and we made sure that the plant itself had no contact with the water, so only the roots were hanging in. Now we would put a floating chamber, or a chamber, it's not floating in this case, but a chamber on top, and we would measure the methane emissions. And we could vary the methane concentration in the water. We could also play with the biomass of different plants. We had different temperature treatments. Now we could control everything. And very interesting, we found a very strong linear relationship, even across the different treatments, with dry weight of the plant. So the bigger the plant, the higher the emission. Maybe not so surprising, but anyway, it was very nice to find this. Then when we correct for the biomass of the different plants, and we plotted it against the concentration that we had in the water, so the concentration that the roots were exposed to, we found also a very strong relationship. So the higher the methane around the roots, the higher the emission. Also maybe not so surprising, but it was surprising to find this very strong relationship. Then we went back to the field data, and we plotted it also against this concentration. So it was either the concentration in the water around the roots that we measured in the surface water, or in the case that they were rooting in the sediment, we put the pore water concentration here, which we also measured in the field. And now we find a very nice relationship, also a very strong linear one. Of course, we have this gap between the ones that we measured in the field and in the laboratory, and it's very noticeable that the emissions in the field were much lower than the ones we found in the laboratory. And although we cannot be 100% sure, we attribute this to the methane oxidation that is occurring on these roots We know that these roots leak a lot of oxygen in the water. They have a very high radial oxygen loss. And we also know that methanotrophs are living on these roots. So actually the concentration that the roots are seeing is much lower than what we measure in the bulk water around these roots because of this thin microfilm of microbes very likely reducing the methane concentration just on the edge of the root. So this was, I think, a very nice way of combining lab work with all the field work that we had been doing. So that is this part. These roots really play a very important role in mitigating greenhouse gas emissions. And we made a small model. I mean, we took little parts of the roots and we incubated them in bottles to see what the methane oxidation potential was. And then of course, we also have the emissions. And we made this kind of donut plot. If you look at the inside of this donut, we see that we have a methane emission and a much higher methane oxidation. And this is very simply the per square meter biomass of these roots multiplied by the methane oxidation per gram of root, and then also the square meter emissions that we found. In the outer root, you then see that this methane oxidation occurs mostly on the roots and only a little bit in the water, and that in this case, most of the emission was diffusive and not ablative, because the abolition got caught in the bubbles, or in the roots, sorry. So how does this play out on a landscape scale? I've been talking a lot about methane, but now I would like to make a small detour to CO2, because we also have a very nice data set on CO2, and that's in the Brasil das Águas project. Maybe you have heard about this. There was a couple, Gerard and Margie Moss, that had this crazy plan, and I'm very sorry I was not aware of this plan before it was executed, because it was really great. They went with this hydroplane in only the Amazon regions to more than 400 different sampling sites. They did not only do the Amazon, they did the entire Brazil. I only worked with the Amazon part of the data set, but it's an incredible amount of data that they gathered. They took samples of everything. And we looked at the CO2, and here you see this amazing plot. So they went to places that had not been sampled before. And they took water samples on which we did DIC analysis, and we also had pH measurements, so we could calculate the PCO2 of these waters. And what we found confirmed mostly what has been published by others as well, that during low waters, so those are the circles, the concentration tends to be lower than during high waters, and then I have the triangle upwards. So this has been found by others too, and it's very easily to explain that when there are high waters, of course, there's a lot of connectivity with the terrestrial area and a lot of organic matter coming in that is decomposed, and also a lot of CO2 from the surrounding areas flowing into these rivers at that point. We also found that if you look at the sub-basins, emissions or concentrations in this case, were higher when you have a lot of floodable area in the sub-basins compared with those that had very low floodable area. What was most surprising to us, however, was that we also found tributaries that were largely undersaturated with respect to the atmosphere. So we know that a lot of these Amazonian rivers are very strong emitters of CO2, but as you see the blue dots, then they were mostly measured during low waters, when potentially there's also primary production in these smaller rivers, was below saturation levels, and they also occur exactly in the places where the organic carbon concentration in the soil, which is the background that you see in this image, is low. So although this spatial map really shows or confirms what have been found by others, I think it's really nice to see this spatial variability. And it also shows again that measurements done in one place are likely not to be representative of other places, particularly not in such a diverse area. So here I depicted the areas that we found to be undersaturated. So now I want to switch back to the Dutch contest again. So I'm from the Netherlands. It's a very tiny country. We have 150 kilometers by 300 kilometers, and we have almost 19 million people living there, so it's a very densely populated area, and a considerable part of the territory looks like what we see here. It's a very organic soil, so it's a peat soil which has a lot of ditches in it to drain the water out to make it suitable for agricultural use. These drained peatlands are responsible for a very high share of the CO2 emissions of our country, and hence it's a problem. Not only because of that, also because we're draining this area, it's oxidizing and we are sinking. In some places we're sinking more than 1 centimeter per year. And we all know that the water level is rising and our country is already below sea level, so I had the advice to buy some territory in Germany already from various people, and when we have the money, maybe we may actually consider doing that. But it's not very sustainable. And while many people were looking at the emissions from these terrestrial parts, nobody had been looking a lot at the ditches and the emissions that we found there. And that is where we came in. So we measured the emissions from 10 different ditches around the year and the monthly measurements. So we already had some measurements from summer, and people said, okay, if we do that times, if we consider that in summer is happening everything, we just multiply by that by 0.5, and now we have kind of the yearly emissions, like the average monthly emissions. Well, when we look for abolition, we indeed see that most of the abolition is occurring in summer. So the graph we see here is based on normalized data. We have 4 seasons. We have the green line, which depicts like the average emission over the year. And the gray area is the variation between the different ditches that we measured. And you really see that during summer and autumn, the emissions are highest, which we saw before also because it's very temperature dependent. The diffusive emission is much less seasonally pronounced. I would say you see peaks, which may be related to rainfall events, but there's not really a clear seasonal pattern. For CO2, however, we found almost the opposite as for methane abolition. The CO2 emission is highest in the colder seasons, autumn and winter, which is not so surprising because now we don't have a lot of primary production, and there's also an excess of rain, so there's a lot of terrestrial CO2 and organic matter coming into these ditches. What our measurements also learned is that our emissions are at least 2 times higher than the emissions that are reported in the IPCC guidelines. So we are champions in ditch emissions, I would say, which is very unfavorable, because if we scale this up, and we have 300,000 kilometers of ditches in our country, so 1,000 times the length of our country is ditch, then we simply multiply this by the emission factor that we found. We know that 7 to up to 16% of our methane emissions from our territory comes from these ditches alone. So here we did not even include the lakes and the rivers that obviously we also have. So it's really a high emission. And then we did not even account for these things. And this happens every other year. Each farmer has to make sure that the ditch keeps a certain level, and somebody comes to check if this is actually the case. So they are legally obliged to keep their ditches deep enough. And what farmers do in this case is they have these kind of devices. They suck up the sediment that is there and they spread it out over the land. And of course, this also comes with huge emissions. What is the happy side of the story is that the emissions could be even much higher if it were not from the methane consumption of the microbial community in these ditches. Also here we looked at the methane oxidation on the plants that we found in these ditches, and some plants are really fully infested with aquatic plants, as you can see here. And what Quinten and Romee did is they incubated these pieces of plants, the roots and the shoots, in bottles to see what the methane oxidation potential would be. And then the same as in the case of the Brazilian water hyacinth, we simply looked at the percentage of methane being oxidized on these plants. In this case, there's also very little methane production in the water, but a lot of methane oxidation on different plant species. One plant species was much more abundant than the other. And in this case, we found that the emission was 99% caused by abolition. So I think this methane oxidation, imagine if the methane oxidation was not there. We would find a factor 2 higher emissions very likely from these ditches, which is already very high. So this is a very strong ecosystem service that these plants and this microbial community is providing to us. I don't know if you like Gouda cheese. Gouda, that's the way we pronounce it. But this cheese has a very high carbon footprint. As we have just seen, this is grown on these types of meadows. So next time you eat a piece of cheese, please remember the impact on the environment. So far, this has not been included in this footprint analysis, nor in our national greenhouse gas reporting, but this is changing. We are in touch with the national reporters, and from this year on, so reporting over 2023, we're also going to include the greenhouse gas emissions from ditches. So switching back to Brazil. So we had these human-made systems in the Netherlands, but we also have human-made systems worldwide, obviously. And we are here in Brazil, we're working in Rondônia and in Minas Gerais in different fish ponds. Fish aquaculture is on the rise, and although I really personally prefer to go to the Pantanal and do measurements there or in the Amazon, I think as limnologists we have a lot to contribute also in these man-made systems such as ditches and aquaculture ponds, which are obviously less enjoyable and less beautiful than the other systems. But I really think here, because of all the management options that we really have a role to play. And aquaculture is booming, as we can see here. Until some years ago, the footprint of cultured fish was mainly based on the greenhouse gas emissions associated with transportation and with the feed production for the fish. And nobody was looking at the greenhouse gas emissions from the ponds itself, which can also be considerably. So that's what we set out of doing. Now there's quite some work also done in Asia, particularly in China, and we are working on the Brazilian version of that, not only looking at the ponds when they're wet, but also considering the dry phase when the ponds are emptied. So here I have an example of one of the ponds in Rondônia, which was sampled by several PhD students here from the Federal University in Juiz de Fora and also UNEMAD, where they measured the different greenhouse gas emissions from different types of systems, so different species, and also different management techniques. Because we want to look what the most important processes are and ultimately also define strategies to minimize this. I remember that with Sally we were talking about the emissions and she said just put a chamber on it, but on this type of fish it will be very hard to put a chamber. So what we found so far is that, the footprint of fish was classified as being lower than pork. Of course, then when you add the greenhouse gas emissions from the ponds itself, it will increase and it becomes quite higher than that of pork, but much lower still than beef. There is still a very high variability in emissions from these ponds, so sometimes the footprint is much higher than what I show here. But it also shows that there's a lot of room to play and a lot of options to reduce these emissions from these systems. One of the options is to prevent overfeeding, because in a lot of systems, the farmers, some farmers are very really know what they're doing, and other farmers are just at the start of getting into aquaculture, and they don't know yet how to optimize the production. In this farm, for instance, they are feeding multiple times a day and not too much at a time, but many farmers also throw in a lot of food one time a day, and then the oxygen concentration strongly drops, and this is very likely when we have the highest emission. Another thing that we show is that when there is little sediment, so when after every cultivation period they remove the sediment, the emissions are much lower. There are also promising insights from oxygenation, which of course could reduce methane production and also enhance methane consumption. So these are all things that we're looking at to try to minimize the greenhouse gas emission. Of course, there's much more to it when we think about aquaculture than just greenhouse gas emission. We're also looking at nutrient budgets, because of course we would like all the nutrients that we throw in to end up in the fish. And this is preliminary work, so don't pay too much attention to the numbers that we are putting here, but we are building a data set to look at different types of systems and see where the input is ending up. And what we for sure are already seeing is that a lot of the nutrients that are thrown in end up, in the sediment and also in the water that leaves these ponds. And this is of course not what we want, so there's a lot of optimization to do there as well. We are not aquaculturalists, so of course we are working together with people that are specialized in aquaculture. We focus on the greenhouse gas emissions and also on the downstream eutrophication, but we team up with animal physiologists and also social scientists. And the ultimate goal is, of course, to remove the footprint, but also to enhance the living conditions of these fish farmers. And here I would particularly like to mention also the PI on this project, which is Nathan Barros, who unfortunately could not be here today, but he plays a major role in this project that I just presented to you. So the last part of my talk, I would like to go into some mitigation options of greenhouse gas emissions. I already mentioned a few, talking about the ditches and about the aquacultural ponds, but there are many more. And together with José Paranaiba, who's also sitting here, we wrote a review that we submitted to Inland Waters, which is associated to my talk today, to look at all the different mitigation options that have been considered in the literature. And we focus mostly on greenhouse gas emissions, but of course there are win-win as well as trade-offs with other ecosystem services of many of these measurements or measures that we discuss. So we already discussed oxygenation of fish ponds as a possible mitigation measure, or dredging of these fish ponds. But we found many other, and we developed a matrix looking at what the target of the measure was. So, for example, minimizing methane production or optimizing oxidation. And the type of measure. So is it a physical measure, a chemical measure, or a biological measure? So I'm not going to talk you through this whole matrix, because we found that actually a lot has been investigated already, with different levels of readiness to be applied. But I will guide you through some examples. So when we think about reducing the compounds of fueling greenhouse gas emissions, of course we can think about land use planning in such a way that we don't have too much erosion. Everybody that went to the Cascades in the last couple of days realizes that erosion is an enormous contributor of soil and organic matter to the water column. So if we can do a better land use planning to reduce this eventually will also reduce greenhouse gas emissions. There's also quite some work done by Brazilian groups on strategic hydropower planning, which, depending where you put your dams, can really enormously contribute to the reduction of greenhouse gas emissions per kilowatt being produced. You could also think about dredging or more, what's the name again? Geoengineering options when your external loading is already tackled, but you still have a very strong internal loading. These could also be options that you can consider. And that is something we looked at also again in a mesocosm project. What we did is we compared different measures, dredging or adding a P-binding agent, in this case lanthanum modified bentonite, to fix phosphorus. And what we saw is that dredging strongly reduces the methane emission, which is not surprising because you could have less substrate to produce methane. But this P-binding only reduces methane emissions the second year. And that is because after this addition, we found a shift in plants. Now, we don't have floating plants anymore, but more submerged plants because the nutrient concentration in the water considerably dropped. And then you get this effect that I explained to you earlier. Of course, when you dredge, you also have to think what is coming, what is the fate of your dredge material and what is coming out of that. So you really have to have a life cycle analysis of this, and that we're also working on it, because it really depends whether you spread it out over the land or you put it in a dredge depot, as you see on the picture here. So another thing that we did in collaboration also with Michael Lurling, who is also in the audience today, is that we looked at the effect of iron chloride addition, which is also applied to bind phosphorus and to combat eutrophication. When we compared the mesocosms that were in this pond around this very nice castle that you see here, this pond is suffering from cyanobacterial blooms, so the water authorities wanted to do something about that. In some mesocosms, nothing was done, and in others, this iron chloride was added. And we really see a strong reduction of methane emission from the treated mesocosms. So this was in part also related to the shift in plant composition, but there was more to it. We looked at the microbial community in these sediments, and we found that they were very comparable between the treated and the untreated ones. But if you look at the methane oxidation potential, and here we added labeled methane and we measured back the labeled CO2, you really see that the blue line, which is the treated sediment, has a much higher anaerobic methane oxidation than the control. And this shows that in this case there may also be a potential win-win situation. This iron chloride was added to the mesocosms to combat eutrophication, but in the same time we also found a lower greenhouse gas emission, which was very nice. Well, as a very last example, and which may also be a very controversial one, is that minimizing greenhouse gas emissions can also be related to enhanced salinization. And whereas salinization is often seen as a negative effect, I think it's unavoidable that our country is going to be more saline in the future. As I told you, we're sinking, the seawater level is rising, and we have to flush out saline seepage that is coming into our systems continuously. If we have a very dry summer, we don't have enough fresh water to do so, so we become more saline. Some water and nature managers actually are embracing this, because some wetlands used to be brackish and we flushed out the seawater to make them fresh. So they are thinking about resalinization of their wetlands. And this leads, we had a very long-running mesocosm experiment, and we see also that this can also have a potential win-win situation with the methane emissions. Of course, farmers and the environment in this region are not yet ready. It would mean a significant shift in crop choice for them. So most of our work, I have given some examples. We have mostly worked with macrophyte management. And I think, and I hope I convince you that these plants, with the associates microbial community that they have, are very important players, especially these submerged plants that have a high radial oxygen loss. I think for many of the systems, at least in the Netherlands, if we manage to get them back, we will have a less strong increase in greenhouse gas emissions. So restoration measures in this sense could really have a win-win situation. For the Water Framework Directive, we need to target them, and it will also lead to a lower greenhouse gas emission. So as to conclude, I think it's very clear that aquatic systems emit large amounts of greenhouse gases. This is strongly on the rise due to all kinds of human impacts, but mitigation measures are gaining attention. And I think they're promising from multiple perspectives. So from the ecological perspective, because a good water quality and a high ecological status of our waters combine with low greenhouse gas emissions, but also from a sustainability perspective. We want to reduce the footprint of our food, for instance. So this is what I wanted to share with you. And obrigada pela sua atenção.